Greeting examiner, my name is Demi Ivan and I'm going to present my final year project proposal presentation. My title is Genetic Transformation of Mungbean with sago palm chitinase to cDNA. Introduction. Mungbean, or the scientific name is Vigna radiata, is one of the most important legume seed families. It can be consumed as seeds but also as a sprouted form of celled vegetable. The seeds of the mungbean contain protein, carbohydrate, and dietary fiber. Genetic transformation. Genetic transformation of animals and plants can be carried out by using the methods such as particle bombardment, gene gun, and electroporation. In this study, Agrobacterium tumefaciens is used as the genetic transformation in plants, whereby in plant transformation, the bacteria will infect the dicot plants such as mungbean, whereby it will infect the wounded sides of the plant, whereby it can cause crown gall disease to the plant. Problem statement. Firstly, can the sago palm be expressed as a sago palm chitinase 2 cDNA managed to express and integrate into the mungbean genome system? Secondly, can the plant transformation of the mungbean be transferred using the agrobacterium-mediated gene transformation? Third, research question. In gene transfer of plant transformation, thus the agrobacterium-mediated transformation method is effective method in transferring the gene of interest into the genome of the plant system, which is the mungbean. The objectives of this study is, firstly, to detect the sago palm in the mungbean genome. Secondly, to insert the sago palm chitinase 2 cDNA into the mungbean genome by using agrobacterium-mediated transformation. And thirdly, to develop the transgenic mungbean plant so that it will be resistant to the common diseases such as fungi disease. Literature review. Mungbean is one of the major grain legume that is widely grown in developing countries such as Africa and South Asia. It is one of the source of protein nutrition to human as well as to feed the livestock. One of the advantages of cultivating mungbean is to improve the fertility of the soil by undergo the nitrogen fixation. There are many studies involving genetic transformation of mungbean that is mediated by the Agrobacterium tumefaciens. Genetic transformation mediated by Agrobacterium tumefaciens. Genetic transformation is a process whereby the genetic material or the gene of interest is expressed into the certain organism such as plant or bacteria. Genetic transformation is one of the major methods in expressing genes of interest into Agrobacterium tumefaciens is one of the major methods in expressing gene of interest into the desirable plants and to get the expression into the plant genome. This is because Agrobacterium has the natural ability to infect the plant that is injured. In the Agrobacterium tumefaciens, there is Ti plasmid, whereby in the Ti plasmid there is transfer DNA where it is responsible to transfer any gene of interest into the plant genome, whereby it will be integrated into the plant genome. In this FYP study, Agrobacterium tumefaciens strain EHA109, which harboring binary vector PGSA1131 will be used, which is contain sago palm chitinase 2 cDNA. Chitinase. Chitinase is enzyme that is responsible to degrade chitin. Class 2 chitinase is being used as the gene of interest. Plant chitinases have strong antifungal enzyme, which often used in genetic engineering to enhance the tolerance of the plants towards fungal infection. Binary vector is used in the Agrobacterium tumefaciens-mediated gene transformation method is the Ti plasmid. In the Agrobacterium tumefaciens, the binary vector is the Ti plasmid, whereby the Agrobacterium has the natural ability to infect the plant that is injured by using T plasmid to transfer the tDNA into the desired genome of the plant system. Materials and methods. Firstly is media preparation. To cultivate the half seed explant, MS medium is used, which contain the B5 vitamins, solid growth medium, and 1% of sucrose. Secondly is isolation of binary vector, which is PGSA1131131 from E. coli. The plasmid is isolated from the E. coli so that the plasmid can be used to transform into the bacteria, Agrobacterium tumefaciens. Next is the preparation of Agrobacterium tumefaciens competent cells, in which the bacteria must be able to receive foreign binary vector, which is the PGSA1131. By doing so, competent cell preparation is used. Preparation of Agrobacterium tumefaciens competent cells. The competent Agrobacterium is stored on ice before the transformation. The competent Agrobacterium later then are mixed with the plasmid PGSA1131 so that the competent Agrobacterium can receive the plasmid. In the end, the Agrobacterium is grown on media bed, and then the colonies are expected to grow to 100 to 200. In the end, the Agrobacterium tumefaciens strand harboring the PGSA1131 plasmid can be used in the transformation of the Mungbeans. Preparation of the Mungbean seeds, whereby the Mungbean is prepared, such as sterilize or wash the Mungbean before to inject the Mungbean. Before that, the seed will be decoated. After that, one of the cotyledons will be removed. Therefore, it will be left out with the embryo that's still attached to one of the cotyledon, or can be called as half seed explant. The location of the injuries of the half seed explant is at the embryonal axis of the half seed explant. Next is the preparation of the bacterial suspension. The bacterial suspension, which consists of the Agrobacterium tumefaciens strand that will be inoculated into LB liquid medium, is prepared so that it can infect the Mungbean explant that are already being injured. Next is the preparation and cultivation of the explant. The half seed explant that already been decoated and being injured will be immersed into the bacterial suspension that's already been prepared. After that, the explant then will be grown into half strength MS medium, and then the explant will be grown at tissue culture growth room at condition of 25 degrees Celsius with 16 hours of light and 8 hours of dark. It is let to grow until the plant is established, have root system, and then moved to plastic pot that contain soil with the same condition growth. Next is plant DNA extraction. The sample of the Mungbean plant DNA extraction is by taking the leaf of the plant. The protocol is by which is using CTAB method. Last but not least is polymerase chain reaction and agarose gel electrophoresis analysis. For this, the polymerase is done to verify or analysis of the sago palm chitinase 2 cDNA. And then to analyze the PCR product, the agarose gel electrophoresis technique will be used to assess the quantity and quality of the PCR product. This is the PCR component and the PCR cycling conditions. Last but not least is the expected outcomes. Chitinase 2 cDNA will be transferred into the Mungbean genome system, and the method is used to perform the transmission of the Agrobacterium tumefaciens into the Mungbean system is by injuring the embryonal axis of the half seed explants. The presence of the sago palm chitinase 2 cDNA is detected by PCR. This is the expected working schedule for my FYP proposal. That's all from me. Thank you.